So I'll try to tell you how we created the chromosomal translocation in iPS cells to be used as a model for the infant T712 acute myeloid leukemia. And AML is the most common leukemia in adults, but it's also seen in about 20% of pediatric leukemia cases. It's a heterogeneous disease, and it's regarded as a stem cell disorder, and it has in general a poor prognosis. We are, though, interested in childhood AML and particularly infant T712 AML, which is a rare disease, but it's still the second largest subgroup of infant AMLs. It's only seen in children under 2 years of age. It's usually associated with a trisomy 19. There is a fusion transcript, however, it's just seen in approximately 50% of the AML cases. But what all do have in common is the overexpression of MNX1. So the genes affected at the breakpoints are MNX1 and ETV6, and they are both transcription factors. They are involved in development. MNX1 is upregulated in several cancers, whereas ETV6 is a significant proto-oncogene, but also an anti-oncogene or a tumor suppressor gene. And then we have our MNX1 ETV6 fusion, where the function, if any, is unknown today. Most of the studies conducted so far has been done in mouse models, primarily with this bone marrow transplantation model, where you can overexpress the different genes you're interested in, fusions whatsoever. But this is still a human disease, and it would be nice to have a human cell model for this. So we decided that we try to create this. And by using the breakpoints that are seen in the patients, which is downstream of the MNX gene on chromosome 7 and in between exon 2 and 3 on chromosome 12. When introducing these to the cells, we also add in double-stranded oligos that will work like a repair template if you do just a simple knockout in the gene. So these will bring the chromosomal partners together to join where they should and give rise to these derivative chromosomes. Since we want a balanced translocation, we want both of these variants, but even though this is the one that is most interesting to us because that's where we have the MNX ETV6 fusion. Since this is a web or a workshop on simplifying iPS cell modeling, I spend a few minutes on some details in the procedures and to share some tips and tricks that I've found valuable during this project. First and foremost, a robust iPS cell line is very important, and that you have a reliable culture system that allows you to manipulate with the cells and also that can support them as single cells after the manipulations. And I found my favorite system, and it's the ChIP22 cell line and the DEF culture system from Cellartis that has been fulfilling all the requirements that I have. When it comes to the guide RNA, I would suggest that you take the two components separately, and if you can, design more than one CRISPR RNA to really be able to test the efficiency of cleavage in your particular cell line. Then you can also use one common tracer RNA, and if this is tagged with the fluorochrome, you can use it for FACS sorting when it comes to selecting cells that have been transfected. And then for Cas9, I've been working with a protein, which is, you can buy it ready to use in solution. And the advantage of that is that you can make the Cas9 guide RNA complex in vitro already before delivering into the cells. And then you of course need an optimized transfection method, and I've been using the Lonza 4D nuclear effector because electroporation or nuclear is the way to go when it comes to iPS cells, at least in my hands. Then, as I said, if you design or want to check your CRISPR RNAs, it's good to test the cleavage efficiency, and then it's just to run a PCR and a T7 endonuclease assay to see that you really have something happening in your cells. It's seldom 100% cleavage, but at least you can see the bands that you're expecting from the CRISPR activity. When you've decided which CRISPR RNA to use, you go ahead and design your single-stranded oligos, and you also need primers to check for a correct translocation. So when you have everything set and ready, it's time to go, and I will just walk you through my procedure and the outcome of this experiments. So you transfect yourselves, not yourselves, your cells, and FACS sort them. Then you seed out single cells in 96 well plates, and after just a few days you can identify emerging clones in the wells. These you expand, and then you, after weeks, you have enough cells so you can do a PCR analysis for a correct translocation. And this is done by just a simple PCR with primers on each side of the fusion site. And in our case, we want a balanced translocation. We want both of these reactions to have taken place. So you need to see a signal in both PCR reactions. Sometimes you just see one of them and then the other fusion hasn't worked out. So what about the outcome? I started with a bit more than 700 single cell seeded wells, and out of these I had outgrowth of a little bit more than 100 clones, and of these I found 6 clones that were positive for both fusions. This of course had to be validated further, and then we used the FISH analysis, where you have fluorescent probes for, in this case, the 2 wild type alleles, the 7 and the 12, and then we have the 2 derivative chromosomes, which give a signal for both 7 and 12. We also wanted to test that we had retained the pluripotency of these cells, that we hadn't messed up anything else with them, so we differentiated them to all 3 germ layers, and that was successful for all our 6 clones. We also tested or run a SNP array on the complete genome to see that there weren't any larger or any damage induced to them. So far so good, but these are still iPS cells, and we are studying leukemia, and to be able to do that, we need to have hematopoietic cells. And so the next step was then to differentiate our iPS cells into hematopoietic cells. And after testing a few published methods that didn't really work very well, we found this commercially available kit from Stem Cell Technologies, which is easy 12-day protocol with just 2 different media. And we used this for differentiation of 5 of our iPS lines, and also the parental iPS line, the CHIPsy22, then without the translocation. And these were characterized with immunophenotyping with flow cytometry, some gene expression, and colony forming assays. And the resulting hematopoietic stem and progenitor cells from this differentiation protocol is characterized by CD34, CD43, and 45 expression. And we could see that there was not any difference really between the T712 clones and the wild type parental line. And also that around 70 to 80% of the cells were triple positive for these markers. We also analyzed the cells with a broad panel of different markers for hematopoietic cells, and found that we had a pronounced decrease in expression of CD235, which is a marker for the erythroid lineage, and also the CD41, which is a marker for platelets and megakaryocytes. And then the opposite, increased expression of CD45 RA. This is a very broad marker and it can be seen in many different cell types, but among those on myeloid precursors and monocytes. And if we have a look at the hematopoietic tree with the stem cell at the top, and then you have the different lineage differentiation routes to the mature cells that you find in the blood, these results indicate that we have a block in the differentiation here towards the erythroid and megakaryocytic lineage, and a shift towards the monomyelocytic lineage. And this resembles what we can see in the patients, because it's a myeloid leukemia. We have a domination of myeloid cells, and these patients often present themselves with anemia and bleedings. So this fits very well with the model. We also had a look at some gene expression. We could see the fusion there in the TF712 clones, of course, not in the parental because there wasn't any translocation. But we also had a high expression of MNX1 in the T712 clones. It was not present in the parental cells. And this is more or less never expressed in normal hematopoietic cells either. So this has something to do with the translocation. And we hypothesize that they are both coming from the derivative 12 chromosome by alternative splicing, because the fusion contains exon 1 of MNX1 and then the rest of the ETV6 gene. But we can also get a full-length wild-type MNX1 since we have the whole gene translocated to this place. We could also show the MNX expression on the protein level in the lines, and there's nothing in but we could never see the fusion as a protein, and this has never even been published by anyone else. And since we can't see this expression in more than 50% of the transcripts expressed in more than 50% of the patient cases, we believe that fusion in this case is not a player in the leukemia development, but rather the MNX expression. We did some colony assays which showed an increase in the number of progenitor cells, and we also looked at proliferation after the differentiation. We kept the cells in culture for a while and could see a difference in proliferative capacity where the T712s were kept alive and divided longer. So just to sum this up, in short, the features of this differentiated iPS cells. So we have a block in differentiation leading to a shift towards the myelomonocytic lineage. The CFU assays support the increased number of progenitor cells. We have this MNX1 overexpression, can see the fusion transcript, and we see this increased proliferation. And all these are hallmarks of this leukemia. So we do think that we have a promising cell model for this T712 AML. And this was done then within our small research group headed by Professor Lars Palmqvist at Gothenburg University and Sahlgrenska University Hospital in Gothenburg, Sweden. So thank you for listening.